Well, it's at 7 and I've been asked to start promptly. Let me introduce myself. My name is Derek Constable. I'm the technical director at Jubilant HollisterStier, who manufactures allergen extracts. And my presentation today is going to be on that subject, i.e. the manufacture, standardization, and the use of allergen extracts. What I'm going to do is to as I say, there's my disclosure, Eric. I'm a full-time employee of Jubilant HollisterStier. I'm going to split the presentation into 4 parts: raw material and processing, the extraction of the raw material through to a fill finish of the product, and then the testing and release of the finished product, and then give you some background or basic information on how to formulate treatment sets. So starting with the raw material and processing. I'm going to run through the 4 main raw materials that we handle at the facility. These are pollens, venoms, house dust mites, and hair and danders. Just focusing on the pollens for the moment. Our raw material collection is basically collected either from the natural environment, i.e. we go out and we collect pollen during the pollination season, or we're actually cultivating those materials under controlled conditions. Once we've obtained the crude material, they're usually cleaned and dried, used through simple sieving or particle separation. And all these materials, before they can be used, have to meet specific FDA and quality guidelines. In particular, that they contain less than 1% foreign material, and that we've confirmed the species of the pollen by a botanist. As you know, pollens are pollinating throughout the year, and initially you'll see in the early part of the year tree pollens coming into bloom. Towards the middle part of the year you'll begin to see grasses, and then towards the end in the fall you'll begin to see weeds appearing. Most of our collection is, as I say, is done out in the field, in the natural environment. And this is usually done by a vacuuming up during the pollen. We send crews out to areas where a particular pollen is about to pollinate, and we will actually vacuum the material up into crude large dispensers. Alternatively, because of some pollens that are difficult to handle, we will actually pick them and display them in a greenhouse on water, called water setting. And we wait for the pollen to actually come out and drop down, then we can collect it on these little white sheets that you see under the pots. Once this crude material is brought into the facility, we then have to go through a drying process. And we do this by isolating them into individual rooms. We spread them out. The air, the heat, and the humidity is under control. All incoming and exhaust air is filtered, and all surfaces in each of the rooms is cleanable and constructed with non-textured or fibrous material. And in between each of the pollens coming in, we will thoroughly clean and then test that room for the cleanliness. The whole of this is aimed at ensuring there's no cross-contamination between the different species. Once we've dried the material, we will then separated it by sieving, or in some cases vortexing, to remove those non-pollen parts until we have a purity down to about 90% of the pollen. Moving to venom, we have 2 general ways of collecting venom from venomous insects. Honeybee actually is the simplest. We collect this as a pure venom crystal by electrostimulation. And what we basically do is place a metal grid in front of the entrance to the bees' hives. So they have to walk across this grid in order to enter. We apply a very mild electrical current through the grid, which causes the bees to involuntarily sting downwards onto a glass plate that we've laid below the grid. The bees are unharmed, they carry on through and they'll come out again and repeat the process as they fly back. We can then air dry those crystals on the glass plate and then straighten them up for collection and use later on. The other insects that we collect are the vespids. These are basically the yellow jackets, the paper wasps, and the hornets. Now, these are much more aggressive species than honeybees. And what you will find is that if you try to use them by this electrical grid process, they get very angry. They will venomate, but they will also defecate. And so you have a complete poisoning, if you like, of the venom material. So what we have to do, we send out collectors around the country who harvest the nests and recover the insects. They're frozen as soon as they're collected to preserve the venom inside them, and then they're shipped to our facility for further processing. We will inspect them to make sure that they've arrived in a frozen state, that there's no more than 5% males, as you probably know, is males have no venom sacs, and there are no extraneous materials. We inspect a few of the insects on each of the batches that come in to make sure that the venom is still clear and there's no cloudiness, i.e. that the proteins have not become denatured. The sacs are individually dissected and placed in solution, and these can then be forward processed to isolate the venom components. It probably takes somewhere between 10 to 30 insects to produce a single dose of venom. And by dose, I mean it's about 100 micrograms of pure venom. Moving to mites, house dust mites, these are basically cultured in-house. And what we do, we inoculate medium with a seed culture of these house dust mites. These are grown over a period of time at a defined temperature and humidity, and then we will separate the whole bodies of the mites from the spent medium, i.e. the consumed medium, and they're dried. And you finish up with this fine gray powder that is essentially 99% pure. There's always a little bit of little medium that carries over, but it's extremely low and there's no significance from the processing that goes forward. With hair products, we basically get everything from veterinarian certified healthy animals. These are both dogs, cats, horses. And we don't, if in the case of dogs and cats, we have no restrictions on the type of breeds that can be used, as there's no such thing as breed-specific allergens. Although we do prefer not to have more than 25% of a batch of any one species. When they're received in the facility, they're inspected for foreign matter and appearance to make sure they are coming from the correct species. So we have the raw material. We then move into the process where we begin extracting those raw materials to recover the soluble allergens. And this is done in a fairly similar process for all species, with a few exceptions. We start off with simple extraction process in some sort of medium. We then clarify that to remove any solids and leave behind the solubilized allergen. Some of the extracts are further processed either through dialysis or precipitation, all of which finishes up with this bulk filtered material. We can then sterile filter that solution into our sterile cores. We fill it into product, we stopper and seal, label the product, send it for testing, and eventually release. And in the case of some extracts, which are particularly controlled by the FDA, we have to submit those samples and results to the FDA for them to do ultimate product release. And I'll get into these details later on. Throughout this entire process, there's the quality and environmental checks at all stages to make sure that we're producing this in a clean environment and with minimal potential for contamination or introduction of bacterial components. So let's just focus a little bit on the extraction process. Basically, there are three aspects of extraction. We want to release the allergens. We want to release them into a solution that will instill stability of those products. And we also want to protect those extracts from bacteria. In terms of release, most materials, we rely on a passive elution of allergens into the extraction fluid by stirring at room temperature for several hours. The allergens will be released naturally through the germination pores of the pollens, or are flushed or dissolved off of the surface of the hair fibers. In terms of releasing some of the materials, they need a little bit more encouragement, particularly those that are encased in rather hard cuticles or in the venom sacs of insects. So for venoms, we will split them open using a homogenizer. And this is like a highly spinning steel blade that will actually break open the sacs and release the venom into the extraction fluid. In the case of whole body mites, we actually ball mill these to split open the hard exoskeleton and release the allergens from the insides. The second aspect is stability, and that depends on the choice of extraction fluid you're using. In most cases, we use 50% glycerol. The advantage of the glycerol is that it coats the allergenic proteins and reducing surface-bound water, which helps to stabilize the conformational structure of the proteins. The downside is that 50% glycerol vaccines can be somewhat painful to inject. We also do use non-glycerol containing fluids that can be used, but unfortunately these don't provide as good a stability protection as the 50% glycerol. In some cases, we will actually add a small amount of human serum albumin, which helps protect the stability of these proteins in the absence of glycerol, but it's only really observed or noticeable when you're dealing with very dilute solutions of allergens. And the third aspect is bacterial protection. One of the advantages of using 50% glycerol that it is also has bacteriostatic activity, which means that you're not going to get bacteria growing in the presence of 50% glycerol. And this helps protect the product through the extraction process until we get to final sterile filtration from the growth of bacteria. It also eliminates, if it's desired, the use of other bactericides such as phenol. Because non-glycerol extraction fluids lack the glycerin, we need to add some bacteriostatic protection to it. And this is usually done by adding 0.4% phenol that can be added. It's safe, but it can have a deleterious effect on proteins, especially in the diluted form. And this is sometimes where the human serum albumin comes in useful, since it can help protect those very dilute proteins from absorbing to the glass wall or being denatured. Once you finish the extraction process, we obviously have to clean out the spent medium. And the way we do that is either a first crude separation that occurs, and this is done by a simple filter or a bulk filter that we will take out all the main solids. And then this will move into clarification phase, where we use progressively, we're using membranes and filters of progressively smaller pore size until we can pass the solution through a filter down to about 0.8 to 0.2 microns. At this stage, you've essentially eliminated all the particulates and most of the bacteria from that solution. However, at this stage, it's not considered sterile. As I said earlier, some of the allergens are processed, have some additional intermediate processing steps. This tends to be focused on extracts that are very dilute, and we'd like to increase the concentration. Some companies use a process called tangential flow filtration. This is basically you're passing the extract over a filter that will remove proteins or components less than 5,000 Daltons and leave behind a cleaned up solution of just the allergen proteins. You can also concentrate it in this stage to give you this highly concentrated material. Another process is you can precipitate the proteins from the solution, and this is usually done by sorting out or solvent precipitation. You basically take the extract, add one or the other to that solution, the proteins will fall out of solution, you can then collect them and then vacuum dry the material to finish up with a white powder that you can then use for further processing. Once you've got your solution in a format you now want to fill it into product, we will go through a sterile filtration stage, which is basically filtering the material into through a 0.1 or 0.2 micron sterile filter into our product filling core. The filling core is where we're actually handling the processing. As you can see in the pictures, we're filling them into vials. And this is done under an ISO 5 or class 100 conditions, which is basically telling you the particulate load in that area, which is extremely low. We use high-speed fill lines for large lots of product, but we produce a lot of small lots of some of our allergens. And for these, most of them are done by manual filling into vials with pipettes. In the case of Venoms, we will actually sterilely load them into lyophilizers and then freeze dry those materials to finish up with the familiar sort of white plug in a vial, which is our pure Venom product. These products are then sent down to our quality labs where we will do the testing. I should say at this point, we have, if you like, two classes of extracts. One we call non-standardized extracts, and then the standardized. The non-standardized extracts are basically manufactured on a weight volume basis. So for example, a 1 in 20 weight volume is 1 gram of raw material that's been extracted into 20 mils of extraction fluid. On these products, we will apply the testing of sterility, safety, appearance, and protein content. For standardized extracts that have a US standard of potency, we will actually do additional testing. And from these, we're doing the same tests as we do on the non-standardized extracts, but also we're applying an FDA mandated potency test. In terms of the FDA standardized extracts, this is a limited range of products. This is the complete list that the FDA mandates that we have to standardize based on a potency assay. They include the cat, short ragweed, both of our house dust mites, several grass pollens, and then of course the 5 Venom products that we are using. But bear in mind, there are probably over 200 other allergen extracts that fall under that category of non-standardized, i.e. they contain no US standard potency and are labeled in weight volume. Looking at these potency methods that are used, there are 3 general approaches that are taken. One is that we measure a major allergen content by a radial immunodiffusion test. And I'll go into these procedures later. But for the major allergen contents, these tend to be used on extracts that are characterized by the presence of a single major allergen. And the two that are tested this way are CAT, where we're measuring the major saliva allergen of FeLD1, and then short ragweed, where we're measuring the major AMB1 allergen. There's a modified version of the immunodiffusion test where we're actually measuring a activity of an enzyme, and we use these for the venoms, where we're measuring the two allergens, the PLA or phospholipase A and HLA, which are the hyaluronic acid. And these are extracts where the allergens are characterized as an enzyme. So we use that characteristic to our advantage in the testing process. For extracts that are more complex, either contain several important allergens, the FDA prefers that we use an ELISA relative potency assay. This is an inhibition ELISA assay, and it's applied to extracts such as grass pollens and standardized mites. As I say, I'll go into these tests, these procedures a little bit more detail now. In particular, the radial immunodiffusion or radial diffusion assays, these are basically performed on thin layers of agar into which have been infused antibodies to the allergens, as in the case of the cat and the ragweed, or we put an enzyme substrate into those gels as we would with the venom. We then punch small holes into that agar gel and then create a dilution of a reference of known activity and then plate these out. So you build up a standard curve, if you like, of concentration of the antigen from our standard versus the ring size that from the precipitin rings of the antibody assay, or where the enzyme in the gel has made a clear zone in which you can see and measure the diameter. You plot the diameter of the wells against the concentration, and then you also run your sample. You measure that diameter, then you can simply read off from the graph the concentration of that antigen or the concentration of that enzyme in the solution that you're testing. As I mentioned earlier, some extracts are much more complex, and although we don't necessarily use this type of process for product release, it illustrates to you the complexity of some of the products that we're dealing with. This is a Western blot of a Russian thistle extract, where we're separating the proteins in a gel under the influence of an electrical gradient, and the proteins will separate out based on their charge and their molecular size. And then we will stain it for the protein. So in the upper image, you'll see the protein profile. The lower image is where we've then incubated that gel with serum from an allergic patient who has specific IgE to that allergen. The IgE will bind to those components that it recognizes, and then we can visualize where those antibodies are binding using a tag antibody, and then usually a substrate, a colored substrate that will show up their presence. So you can see in this example, there's at least 6 prominent allergens represented as a total. And for these type of tests, for these type of extracts, as I mentioned earlier, we can use the FDA's enzyme ELISA relative potency assay, or ERP. And what the FDA does is that they set up independently a reference extract on which they will assign a potency. And they prepare this extract, or they purchase it from one of the manufacturers, and they will go out and skin test this extract in sensitive patients. They basically enroll 15 to 20 of these patients, making sure they're adequately sensitive by a puncture test. And they're looking for those that give big erythema diameters greater than 80 millimeters on a reaction to a standard test. They will then prepare threefold serial dilutions of this extract, and then intradermally test them in the patient and measure the wheels that are created. And what they're looking for is they're trying to find out the concentration of the extract that will produce a 50 millimeter erythema diameter. And this will obviously vary from patient to patient. And based on that value, they can assign a potency unit, be a biological bioequivalent allergy unit. So what they're basically looking for, they're creating this curve of all the different threefold dilutions and looking at the erythema size that is created. And they can plot this curve. They can then look at the 50 millimeter area and find the concentration, or rather the dilution they had to prepare in order to give that 50 millimeter. And this is an average mean value of all the patients that they've actually studied. They then take that number and compare it to this standard table. So for example, as in the previous example, they were showing a 14.2, I think it was, a dilution series. This would fall in the range of 13 to 15, and therefore they will label their product at 100,000 BAU per mil. If it had fallen between 10.9 to 12.9, it would be called 10,000 BAU per mil. So this is the standard that they're creating. So how do we use this in the facility to release product? Well, basically, they'll send us the standards. They'll also send us a pool of serum from the patient, a mixture of patients, and we will use these two in an ELISA relative potency assay to compare the two potencies. And what we're trying to do is to show equivalence to the standard. So we prepare dilutions of our product and compare them to the standard until we find the concentration that matches the standard. As you know, all assays have some sort of variability. And so what we're looking for is a number that's coming out, a relative potency to the standard that's falling in a fairly limited range. In the case of grass, for example, we're looking for a number that falls between 0.776 to 1.288. And this is the average of about 6 numbers. And if we get the dilution of our product to fall within this range, we can label it 10,000 BAU per mil. This is an example of a potency assay. So what we're doing is we're taking the reference. Well, first of all, we're actually preparing microtiter plates where we've taken the reference extract and coated it on the bottom of the plates so that it will be immobilized. We can then add to each of the wells of the plate a small volume of atopic serum from the patient that contains IgE, which would normally bind to the immobilized allergen in the well. But what we then do is we take the reference, we make serial dilutions of that product, and then place them in each of the wells alongside the same serial dilution of our sample. And then we can plot and look after the incubation stage, where the extract's inhibiting the binding of the IgE to the immobilized extract on the well, we'll see that the amount of IgE binding will be dependent on the amount of allergen we've added. So you get these titration curves. And we can compare the two curves at the 50% inhibition point and show what the difference is. So for example, with the reference extract, we're getting a number of 110 concentration units. And for the sample, we're seeing 100 concentration units. The ratio of those is obviously 1.1, R over S. And then, as I showed from previously before, 1.1 will fall in the range of 0.776 to 1.288, and therefore we can label it at 10,000 BAU per mil. This is probably of more of interest to you than most of what I've just said. This is on how you are going to potentially prepare treatment sets of the bulk extracts and products that you would buy from any of the allergy companies. In terms of formulation of treatment sets, there's generally two approaches taken. The most common is probably the process that's set out by the academy and the college, where we're utilizing the FDA standard of potency or the label claims on the vials to target a concentration of allergen that we want to treat our patients with. There are, however, alternative methods, and the most common of which is where they're using major allergens of the extracts and targeting a particular microgram for the patient. And I do stress the choice is purely at the physician's discretion of which way they wish to take. If you look at the most of the academy and the college information can be found in the practice parameters that they will update and issue on a periodic basis. First of all, just dealing with the alternative methods, the major allergen content, and looking at some of the pros and cons of the procedures. The pro primarily is that if you're just using a single number, i.e. a single concentration of a major allergen, it's a relatively straightforward concept in order to target a maintenance dose based on those micrograms of a major allergen that you've identified and want to make sure that is being adequately treated in the patient. This process is favored largely in Europe, and even under the FDA's guidance, it works well with some simple extracts like cat or ragweed, which only has a dominant major allergen. The disadvantages is that there's no universally agreed references or methods available other than from the FDA for the cat and the ragweed. And because there's no standardized process, you can basically you could assay the same extract in two different assay systems and get two totally different numbers, which makes it very difficult to compare activities between different companies who are using different methods. And of course, as I mentioned earlier, just measuring a single allergen may not adequately reflect the total extract potency. Using the what I like to call the academy or college method, you have universally agreed references and methods available that are distributed by the FDA for their standardized allergy program. And so you are more confident of getting comparable results from whatever source of extract that you're getting that is standardized and labeled in this format. The advantage of the enzyme, relative potency assays, such as GRAS and with mites, that you have a more holistic approach, i.e. you see all the components that are contributing to the total activity of the product. Of course, the flip side of that is that you can't measure the individual contribution of a single allergen within the total extract pool. And also one of the problems of running these ERP or ELISA type assays is that you require atopic serum, which the FDA creates pools, but they're not always easily replaceable and to make them identical from one lot to another. So that always presents a little bit of a problem when we're switching over to a new serum pool. This is the table that I've taken from the practice parameters. It's not intended to be read at this point, but it's a very useful source to go for to look at probable effective doses of the recognized by the academy and the college that you should be trying to target your products at. This not only covers standardized extracts, the mites, the hair, the cats, grasses, weeds, but also the non-standardized products, which are basically described in terms of weight volume. And they will give you targets in which to try and aim at for preparing your treatment set for treating your patients. So let me just go through a process, an example of how you might want to go about preparing a treatment set. And as you know, the objective here is to prepare a customized allergen mix such that a PED, or probable effective dose, of each component in that mix is at the targeted maintenance dose volume. In order to do that, you need 4 pieces of information. One is that you've got to select your probable effective dose for each allergen that you want to include in the mix. And it doesn't really matter what you actually use, whether it's AU, BAU, weight volume, PNU, or microgram. You can apply that target to formulating your product. However, you need to know what the amount or the potency of the bulk extract that you're going to use in whatever choice or designation, whether it's AU, BAU, weight volume. You need to know that for the bulk extract. You then got to decide what maintenance dose volume you're intending to build up to in the patient. Usually, most physicians are targeting somewhere between 0.3 to 0.5. A lot of them actually use 0.5 mils. And also you need to decide what sort of vial that you're going to prepare, whether you're going to put the mixture in a 5 ml vial or a 10 ml vial. Now, this is just an illustration purely to run through the process. So I picked a 5 allergen extract mix that I'm going to make into a 5 ml vial with the objective of a 0.5 ml maintenance dose. My 5 allergens, the bulks I'm going to be using, are standardized Timothy pollen at 100,000 BAU per mil, oak pollen at a 1 in 20 weight volume, a standardized cat hair at 10,000 BAU per mil, a standardized mite D. farini at 10,000 BAU per mil, and a feather mix at 1 in 10 weight volume. So these are all my components. The next stage is to decide, well, what is the PED that I want to target? In this example, I'm just going to use, these are the ranges I got from the table I showed you earlier. For Timothy, the PED is, you need to deliver 1,000 to 4,000 BAU. With the oak pollen, I need to deliver 0.5 mils of a 1 in 200 to 1 in 100 weight volume. As you remember before, it was a 1 in 20 is my bulk. So in other words, I've got to deliver, prepare a dilution that's represented by 1 in 200 or 1 in 100. And I'll go through how this is done in a minute. And similarly for the cat, 1,000 to 400, the Freeney 500 to 2,000, and for the feather mix, 0.5 mils of a 1 to 200 to 1 in 100 weight volume preparation. And for the sake of argument at the moment, I'm just picking the upper end of each of these potential ranges, bearing in mind that the general concept is that the higher the dose you can deliver to the patient, potentially the better the outcome. I do make a note down here that with the feather mix in that table that I showed you, it does not specifically reference feather mix. However, I always use as a good starting point those the general criteria for non-standardized pollens, such as oak. So it gives you a starting point rather than just a complete guess as to what the effective dose may be. So for those extracts where you have the potency in terms of units per mil, where those units can be BAU, AU, micrograms, PNU, whatever, this is the type of approach you want to take. As I mentioned before, you need 5 pieces of information. You need to know what your PED target is, that's 4,000. You need to know what the potency of your bulk is. In the case of Timothy here, it's 100,000. I need to know what the vial size I want to use, it's 5 mils, and my maintenance dose is 0.5 mil. So in order to achieve the selected target in the dose volume to add to the vial, you do this fairly simple calculation. You take the 4,000, you divide it by the 0.5, which will give you 8,000 BAU per mil. This is basically telling you that in the final 5 mil, the concentration of your Timothy needs to be at 8,000 BAU per mil. You then take that number and divide it by the potency of the bulk, and then multiply by the number of mils you're planning in the vial, which will give you 0.4 mils of Timothy bulk. So basically, you've got to take 0.4 mils of this bulk and add it to your 5 mil vial. In the case of weight volume extracts, the calculation is a little different, but you need the same amount of information as before. I'm going to be targeting the 1 in 100 weight volume at 0.5 mils, and my bulk is 1 in 20, and still my vial is 5 mils, and my maintenance dose is 0.5. So to find out how much I need to add to that 5 mil vial, I will take that 20 of the bulk concentration, divide it by the targeted concentration, which will give me 0.2. It's actually 0.2 mils. And then you multiply that by the number of mils you want to add, the mil size of the vial, which will give you 1 mil of oat pollen bulk. In other words, you pull 1 mil from this bulk and add it to your 5 mil vial. And you can put whatever you choose, you can put whatever number you want in here, and it will give you the amount to use. The only warning I will use when using weight volume is to make sure that everything is in weight volume. Sometimes you will get customized bulks that are prepared and labeled as volume. Don't mix these, as they're not the same. And you won't get the same expected concentrations. So try and stay with weight volume throughout your calculations. So applying all these rules to the 5 extracts that I has picked for this treatment set, using the maximum dose that I want to try and target, these are the volumes I'm going to be adding to the 5 mil vial. 0.4 of Timothy, 4 mils of cat, 2 mils of mite, 1 mil of oak, and 0.5 mils of feather. But you will obviously see that the volume is greater than the vial. It adds up to 7.9 mils. So there's a number of other options you need to consider. Option 1, you can keep all the extracts in one vial by using the minimum target dose. So instead of using the max, let's look at the other end of the spectrum and go for the minimum. You can go through the same calculations, just substituting the maximum dose, the PE dose for the minimum, and you then finish up with another set of numbers, which you will find this time is giving less than 5 mils, i.e. they will all fit in the vial. If you prefer to stay with this and just work with the minimum volumes, then you would take that number and just make it up to 5 mils by adding 2.65 mils of a diluent into that vial, preferably 50% glycerol. Or you can consider manipulating those numbers to somewhere between the max and min in order to give you a total of 5 mils. And this intermediate objective I've just given as an example. This isn't specific, but you can also use this to your advantage by tailoring a particular or emphasizing the importance of a particular allergen over the other, particularly if the patient's main symptoms are due to 1 or 2 allergens. You can add more towards the max dose and then back off a little bit on the minor allergens in order to improve that profile mix for the patient. Of course, you could stay with this 7.9 and just split it between 2 different vials and give the patient 2 injections. That can be done if you prefer to stay with max at all times. Just remember that each vial needs to be made up to 5 mils in order to be able to deliver the correct dose in the 0.5 mil maintenance dose. Just one point to consider is that some extracts that you'll be handling have a very high protease activity, that if they're sitting in the vial with other allergens, they'll begin to degrade. And these are proteases particularly associated with molds and cockroaches. And you should consider putting these in a vial by themselves. And basically that brings me to the end of my discussion. If you like, we can go on to, I can unmute you and we can discuss some questions. Let me just. Stop share. Trying to find my unmute all. You can probably unmute yourself if you have a question.
Dr. Constable, do you hear me?
I do indeed.
Yeah, this is Len Altman. Thank you. That was the best description I've ever heard of how we should mix allergenic extracts.
I appreciate that.
I'm glad that the fellows are listening to that. One very simplistic question, are there hypoallergenic dogs?
It's a good question. I think it's more of a myth than a fact. Hypoallergenic dogs all dog species will produce allergen. It just depends on how much. And what you tend to find is that with long-haired dogs, they retain a lot of the dander and saliva from them, and then they give the impression of being more allergenic than other dogs that have little to no hair. If there's very little hair for the allergens to remain adhered to, then they will get out into the environment and shed from the dog's surface. And this is where the concept or the thinking of hypoallergenic dogs come into. But really, there's no such thing in the true sense. It also depends on the sensitivity of the patient. Are you achieving the threshold level of that patient's allergy with the amount of allergen around? So taking those two things into putting them together can give you the impression of hypoallergenic, but the reality is all dogs shed allergens.
Is it true? I mean, the hair itself is keratin, much like our hair. So it's just that the hair carries proteins that come from the skin?
Correct. Or from the dogs salivating over themselves, it then dries. And that's particularly important in cats. They're constantly licking themselves and the saliva then dries on the on the hair, and you pick it up when you stroke them, or it comes off as the cat's walking around. Yes, you're right. The keratin in the hair plays no part in allergy. It's just like a carrier.
What do we know about the longevity of these allergens in indoors? As far as, you know, I've heard if there were cats in a property and you remove them, you can still detect allergen perhaps out to a year.
Oh, yes, it's, well, yeah. Most of these allergens are really quite stable. They're quite robust, and particularly in a dried format. In a solution, as you would in an allergen extract that you would purchase for treatment, you're in an aqueous environment. That creates a lot of issues with stability and denaturation. But in the house, the allergen is dry. And dried allergens, I mean, there's been examples where mold, for example, has been shown to be active, still active after 30 years in a dried format. So they can last for an extremely long time. And that's why the problem with carpets in particular in houses, where the dried allergen becomes embedded deep into the carpet and becomes very difficult to get out. And that's why some people are suggesting you should, if you're really hypersensitive, particularly for house dust mites in particular, you should actually get rid of your carpets and stick to wooden floors that are more easily cleaned.
I welcome anybody else to ask questions. While I'm waiting, I'll ask you one other one. What about the efficacy of mold immunotherapy? It's my impression is that it's not terribly effective, and that we don't very often do it in the Pacific Northwest.
You're right. Molds are a little bit of an unknown in many ways. I mean, clearly people are allergic to mold. Some people extremely so. The lack of standardization of molds makes it difficult to predict good quality targeting of those species. Most of the information on molds have been generated on just 2 species, Alternaria and Cladosporium. And they have been shown to be effective, but the doses need to be very high. As I mentioned earlier, many of the molds have a high active enzyme, a protease activity, and they can, if you like, in a solution, auto-digest themselves. And so the impression is that the quality of these extracts are not as good as they could be, certainly when you're comparing them to something like a pollen or a house dust mite or some of the epithelial extracts. So molds do have an issue. Some people see that they do find benefit from them, and so that's why we continue to have them available. But we can't say a lot about their specific activity, other than I say about for the Alternaria and the Cladosporium, where there's a lot more information.
Hi, this is Paul McBride. If I can ask a question about your dog extract. Are you making any progress on getting the other allergens in the dog extract and moving it towards standardized?
Well, as you know, we can measure the CANF1, and we do also measure the albumin in some of our particular extracts, but I think all the companies do that. The question of standardization is in the dictates of the FDA. They basically tell us what to do. It doesn't necessarily work the other way around in terms of us wanting to standardize an extract. My personal belief is that the FDA would like to see unified standardized extracts throughout the industry. And that's why they started this program and applied it to the house dust mites. It'll be great. We could do it with the dog. We believe we know how we can do it. but the FDA, we can't really do it without the FDA's acceptance, because they're the ones that are going to approve and give us a license for a standardized dog. But I agree with you. I think there's a lot of value in being able to do not just dog, but certainly a lot of other allergens that would benefit by standardization in the wider area than what the FDA has dictated at this stage.
Thank you.
For anybody who has a question but perhaps doesn't want to raise it here, I'm more than happy for you to supply questions by email, and I'll respond to them individually if that would help.
This is Len Altman. I have one other question. How many U.S. manufacturers do we have of allergenic extracts, and what kind of communication goes on amongst the companies to discuss these processes?
There are 3 main allergen manufacturers in the United States that are traditional manufacturers, should I say. There's a number of other companies coming in from Europe, but none of them have approved products at this point. But the 3 traditional companies, we have an association that we meet on a regular basis between ourselves, but also with the FDA, where we can present and discuss general issues that are generally of concern or of interest to the industry with the FDA, and obviously between each other. We don't get into the level of discussing individual company-owned processes, but in general, particularly from the regulatory and licensing and issues with the guidelines and requirements of manufacturing products, this is where there's a great value of being able to talk with each other. And as I say, we do this through the Allergy Manufacturers Association.
So the other 2 companies, Greer, and who's the other one?
ALK.
Okay.
Although it's Stalligen's Greer now.
Well, anybody else, feel free to ask questions. Thank you very much for an excellent presentation, very pragmatic, something that allergists do every day without much thought, sort of the main procedure we do in our practice, and not necessarily taught very well. So thank you.
You're welcome. Which